So what are homeobox genes? Well, they code for transcription factors that signal cellular growth in body segments along the anterior to posterior axis, even before an area has a formal shape. Their products do not initiate cells to become a part of a specific organ. However, homeobox genes activate or inhibit a series of other signaling molecules that will form future segmentation. Signaling molecules such as morphogens like FGF and BMP are directly affected by Hox genes, and they activate genes that alter cell and tissue morphology. Homeobox genes experience a pattern that is simple and linear. This pattern is highly conserved across all animals. From fruit flies to humans, the embryonic gene patterning and expression is very predictable and represents a specific body plan. Hox is simply a contraction of homeobox, which means well-conserved DNA sequence. BMP and FGF growth factors activate homeobox transcription factors, such as MSX1, which guides the development of the proper tooth type. Mutations in MSX1 lead to hypodontia and cleft palate. In the figure shown, the top row shows normal tooth development at embryonic day 14.5 and 16.5 in heterozygous MSX1 mice. However, the following row we can see that the homozygous mutant mice show tooth development that is permanently arrested at the bud stage as a result of the loss of BMP4. How do Hox genes regulate the growth of appendages in flies? Hox genes in insects specify which appendage form on a segment, for example, legs, antennae, and wings. In segmented animals, Hox genes specify segmental or positional identity, but do not form the actual segment themselves, seeing how the protein product of each Hox gene is a transcription factor. There are 8 Hox genes in Drosophila, or fruit flies. They are sequential, and the gene order reflects the expression of each body segment. The gene on the 3' end of the DNA is the lab gene, abbreviated for labial, which is expressed in the head. On the 5' end of the DNA strand is ABDB, short for abdominal B, which is expressed at the end of the abdomen. The knockout of Hox genes in Drosophila causes one body part to develop in another. Hox genes are early contributors in the cascade of interactions that enable the development of morphologically distinct regions in segmented animals. Mutations that arise can cause a leg to develop in place of an antennae on a fly's head. In this slide, we see the conservation of Hox genes among different species and their corresponding regions of expression.
So to answer the question, are teeth considered appendages? The teeth are roughly considered appendages. They're appendages of the oral cavity. The definition of an appendage is a part of an organism that is attached to the main body. They usually serve a specific function or role within that organism. These appendages are specific to the oral cavity as they protrude out of the gingiva and the alveolar bone. They're a part or an appendage that is connected to something larger to make up the whole organism. Skin and hair are also considered appendages. They're also shed as well. These appendages usually immediately replace themselves with a new layer that is underneath. Teeth, however, cannot, unless it's a primary dentition to permanent dentition, then teeth just have only one replacement. So are other human appendages shed? Well, to answer this, yes, other appendages are shed, such as like the hair and the skin. These appendages, so to speak, are immediately replaced, though, by a new layer or a new hair that was growing underneath them. Other appendages like the arms, fingers, legs, foot, those are not shed. And teeth, although them being able to shed, they're still considered appendages. It's a part, they are a part of our bodies, and helps to make up the whole. And it extends out from the gingiva in the oral cavity. So it would be an appendage of the oral cavity. So comparing the tooth and ectodermal appendage development. Well, development for both the teeth and other ectodermal appendages began as ectoderms that turn into placodes and then into buds. So they all start from one basic fundamental root point. They both receive signals from the mesoderm and mesenchyme to differentiate into the various parts after they have reached the budding phase. This is where they specialize highly, and these are where you see the main differences when they differentiate into these specialized forms. They're given different signals, and they use these to develop into their specific roles or functions that they're supposed to be playing. The cells at this point, they're highly specialized and to their specific task, and they're different from one another, but they have the same roots that they started from, like I was saying before. So they may be a hair follicle or a tooth, but it all started from an ectoderm that differentiated down until it reached one of these points.